Hello, and thanks for joining us today for the latest Technology Networks webinar, Testing on Humans, How to Predict Hepatotoxicity and Drug Clearance Ahead of Clinical Trials Using Liver on a Chip. I'm Brian Manning, US Head of Sales for CM Bio, and I'm here to moderate the event. I'm here to have, pleased to introduce Dr. Tomasz Krzyzewski today as our presenter. Tomasz has been Director of CM Bio with over 10 years experience in molecular and cellular biology research. Prior to joining CM Bio, he worked at Imperial College in the Department of Life Sciences, studying immune cell development and stem cell differentiation. He has significant experience in advanced 3D mammalian cell culture, organ-organ crosstalk, and immune cell regulation. In 2017, he was promoted to be head of biology at CM Bio and responsible for biological model development and collaborative research projects. A warm welcome to you, Tomasz. Following the webinar, we will have a Q&A session and we will welcome any questions that you may have. You may submit questions for the Q&A session at any time during the presentation. To ask a question, you should enter your question into the box on the right-hand side of your screen and click send. We will answer as many as possible during the time available. For any questions we don't have time to get to, we will be sure you are contacted offline with an answer. If you experience any technical issues, please click the chat icon in the top right-hand corner to request support. Please remember, you can ask questions at any time, even whilst watching the webinar on demand. So without further ado, I will now hand over to Tomasz.
Thanks, Brian, for the introduction. Hello. So today I'm going to tell you about how we use our liver on a chip technology to predict hepatotoxicity and understand human drug clearance. As I'm sure we're all aware, developing new medicines remains a costly and time-consuming activity, often taking decades and costing billions of dollars. And the principal reason for this is that most compounds that enter clinical development simply do not get approved as new medicines. On average, only 1 in 10 new drugs that enters the clinical trial process ends up becoming a new approved medicine for patients. Now, drugs fail in clinical trials for many reasons, but often this is due to issues with safety and with efficacy, and safety being the principal cause of problems in phase 1 and phase 2 clinical trials. What this data tells us is that really we need better ways to do preclinical drug discovery and drug development so that we can create better therapeutics and better compounds that enter clinical development to give better chances of success. Now, the modern scientific researcher, when they're doing preclinical drug development, has a really wide range of tools available to them. Traditionally, the tools that people would have used would have been traditional 2D cell culture, cells cultured in dishes, or in vivo animal models. And the combination of these two types of approaches has led to the issues that we have today in clinical development, because these two approaches really don't predict the way that humans behave and the way that drugs will behave in people in clinical trials. So a number of more modern approaches now exist to complement those traditional approaches. So in silico, computational based approaches now are allowed to design and predict how drugs will perform without even having to go into a laboratory. Cells can now be cultured in 3D and in organoid type structures, which are much more advanced than 2D. And they're more advanced than that. There's been a significant rise in the use of organ-on-a-chip and microphysiological system technology, which are much more advanced in vitro technology to culture cells in 3D in the lab. And at CM Bio, we've been building organ-on-a-chip or microphysiological system technology for almost the last 10 years. And we now have built the advanced PhysioMimix organ-on-a-chip technology. And this has really been built on technology that we originally licensed from MIT and the laboratory of Professor Linda Griffith. And the PhysioMimix OOC platform consists of open well plates that are driven by our control unit. And the open well plates that are used in this system come in a number of different shapes and forms for culturing different cell types to create different single and multi-organ organotypic models. Now, the way that a researcher can use this technology is very varied. As I say, we have a number of different consumable plates that interface with our technology for culturing different cell types. But even for a particular plate type, multiple different cell types can be used to create cellular models. So we can use everything from basic cell lines through to primary cells, stem cell derived materials, or even whole tissue slices that have come directly from patients. And the key principle of what the system does is no matter what cells we put in, the system is designed to keep those cells alive for a number of days. So typical experiments in this OOC MPS platform is that they're cultured for a number of weeks, typically 2, 3, 4 weeks. And during this time, the cells are continually bathed with media, and that media is constantly perfusing by the microfluidics that are embedded within our plates. We can potentially induce a disease state within the cells while they're in this platform. And in particular, we think very hard about the way that cells are exposed to drugs or therapeutics whilst they're in the plate, so that the exposure that the cells receive is as physiologically relevant as possible and as closely mimics the type of exposure that those cells would receive in a human being. And at the end of any experiment, we can do a really broad range of different endpoints to understand cellular behavior. So we can do complex omics analysis, we can do a wide variety of microscopy, and we can look at clinical biomarkers that are relevant, so we can get the most translatable data from these studies to predict forward what would happen in a human. So let's look in a little bit more detail at our human liver model, which uses our liver MPS plates. So our liver MPS plates have 12 open wells, and each of these wells is an individual unit. So it individually perfuses. So here you have a cut through of an individual well. The media constantly perfuses around the well, and there is a three-dimensional scaffold held within one end of these wells. And this three-dimensional scaffold is constantly perfused by media throughout the experiment. So media constantly perfuses around the well. And what we do is we take single cells. Typically, these are primary cells of hepatocytes and non-parenchymal cells, but they can be other cell types too, as already mentioned. And over a number of days, these cells go from a single cell suspension to form three-dimensional microtissues, an array of microtissues within this scaffold. And the cells, once they're in this microtissue state, have the same microarchitecture and organotopic nature of the same cells as they have in vivo. So they produce tight junctions, they form biocanaliculi, and have a complex three-dimensional nature. When the cells are in this platform and through the constant perfusion of the media, what we create is a really long-term and highly functional liver culture. And traditionally, when you compare to more simplistic 2D cultures of equivalent cells, the cells of this platform are much more highly functional and outperform. So what we can see is we can look at a number of wide variety of biomarkers that are produced from the liver. So for example, liver proteins that are produced, these are very consistently produced throughout the cultures. We can do a variety of microscopy to look at the way in which the cells form. And from a metabolic perspective, we see very high metabolic activity, looking at a wide variety of enzymes. The system is also very transcriptionally stable, and we see gene expression patterns consistently produced by the cells throughout the course of an experiment. So with this liver system, we can do a really wide variety of different activities, and we can utilize it for a variety of different problems. So we can use it for example, for disease modeling, looking at a variety of different compounds, whether they be small molecules or large molecules. What I'm going to tell you about today is how we use the system for both understanding drug metabolism and how we also use it to understand drug safety and toxicology. So with a highly metabolically active system, we have a really broad expression of CYP enzymes, cytochrome P450s. And we've done a number of studies showing that by putting the same hepatic cells into our system and compare them to traditional two-dimensional non-fluidic cultures, we see significant greater levels of drug metabolism in our system. And here we show where we have increased levels of drug turnover of a wide variety of different compounds. We've looked at the effects of how individual donors in our system will vary subtly due to their different expression levels of CYP enzymes, and we can take data sets from our system and using PK modeling, we can then extrapolate forward to how a particular compound will then be metabolized by a human, and we get very close correlation between convert the metabolism of drugs in our system to what is known in clinical studies. We see this for a wide variety of compounds here with different rates of clearance. We can also use our system to look at metabolite identification, and our system has a particular advantage that it has a relatively large volume in each individual well. So what we can do is repeat sample from individual wells across time, and then in combination with sensitive LC-MS analysis, we can then detect for the presence of multiple metabolites from every single well. So here I'm going to show you an example for acetaminophen, paracetamol, and this is the breakdown of paracetamol by the liver. So it becomes a number of different metabolites, and we can detect all of these in our system, and we can detect them across time and how each of these metabolites gets produced at varying different rates. For certain molecules, there are very human-specific metabolites, and using human cells in our system, we can detect those metabolites. And then in particular, we can take enough sample whereby we can look at both cell health and the production of a particular metabolite in the same sample, so we can then correlate if there is a production of a particular toxic metabolite, when that will then potentially have effects on cell health and viability, and what point that would occur. We can use the system to look at very slowly cleared compounds, and these are typically very challenging to study using in vitro cultures because we have a very stable long-term system with high levels of metabolic activity. We can put a variety of compounds in, such as those compounds cleared very fast by the system in just a few hours, and some compounds that take very long time. So again, we can take multiple samples from the same well that remains untouched. We can take multiple samples across time and see the slow clearance of those compounds in our system, which is something that's typically not been able to do using normal in vitro cultures. So I've shown you how we can use our system for drug metabolism. So now let's show you a little bit how we can use it for looking at safety and toxicology, in particular how we can understand and measure drug-induced liver injury. And this is a really key area of research. Drug-induced liver injury is one of the most common reasons that drugs fail in clinical trials for safety reasons. The way we use our system is we can look at both acute and chronic effects of drugs on our liver cultures. We can look at, and particularly what we typically do is not just look at single endpoints for a particular drug. We will look at multiple clinically relevant endpoints: the production of albumin, the production of ALT, AST, the production of urea, and the combination of all of these molecules, we can then get a profile of how this molecule behaves. And here I'll show you a heat map where we've took a variety of different compounds, different concentrations. What we see is a sort of clustering of toxic compounds. They cluster together with a similar profile where we look at a variety of different endpoints compared to non-toxic doses of these same compounds. And we can use the system, again, to not just look at the DILI effects of small molecules, but we can also look at large protein therapeutics. We can look at genetic reagents like assays or viral vectors, and really understand how these can cause DILI in both an acute and a chronic manner. And we've done this with a really wide variety of different compounds of different nature. We can go one step further and we can look more deeply at the mechanisms that occur in drug-induced liver injury in our system. So, for example, because we have very highly metabolically active systems, we can look at the production of toxic metabolites, such as what I showed you earlier for acetaminophen. We can look at how mitochondrial behavior changes over time, and we can even look at cholestatic injury. So, for example, here you see with increasing concentrations of the compound, the production of the particular bile acid is reduced over time. We can profile a wide variety of bile acids in the system and look at their changes over time. Additionally, we can use proteomic style approaches to really understand the broader spectrum changes that occur to our liver cultures when they are dosed with a compound. So here I'll show you where we have looked at the secretome of the cells in our system with and without the addition of a DILI-inducing compound. And what we see is there's lots of proteins that change after the addition of the compound. Lots of these proteins being produced are associated with apoptosis and cell stress. And we can then compare and contrast these to different doses of the same compound or between compounds to really interrogate what mechanisms are occurring following the dosing of cultures. And then we can extend this a little bit further and ask some even more complicated questions using our system. So we, as well as having single liver MPS systems, we also now have multi-organ MPS systems where we can connect the liver to both the gut and the lungs. We can connect the liver to immune cells. And what this allows us to do is to study inter-organ crosstalk. And a recent publication by our collaborators at MIT has demonstrated how this can be done. And with the presence of immune cells either in a single or multi-organ MPS platform, we can look at how inflammation can mediate toxicity. This is a really key area and often very difficult to study using in vitro platforms of just a single cell type. And we're also able to start to think about how MPS platforms can stratify different patient populations and how that will affect their DILI susceptibility and toxicity profiles. So what we have done is taken a model of fatty liver disease, and we can see how when the cells are, so you can see here labeled as lean or fat at the bottom, when cells are cultured in a fat environment to mimic the early stages of fatty liver disease, the cells in the fat environment are much more susceptible to toxic doses of particular compounds, and we see this various different examples. And these kind of observations mirror what is seen clinically, but it's very difficult to then go and interrogate and understand. And so by setting up the platform in a number of different ways, we can really ask really detailed mechanistic questions about the causes of toxicity, and potentially some of this data can inform sort of clinical trial design and patient stratification to think about what toxic doses might be when you take them into the clinic. Now, we believe these two areas of research are very important, and this concept is backed up by our collaborators at the FDA and the group of CEDA, who we have been working with and collaborating with for a number of years now. And we've been working with them to focus on these two areas of human drug clearance and drug-induced liver injury as really key areas and important context of use for liver MPS platforms. And for those of you that didn't see it, our collaborator and my good friend, Dr. Alexander Ribeiro, did a previous webinar for us on this topic, and I encourage you to go and view this through our website if you haven't seen it before. And certainly we see that the use of liver MPS systems in these places is incredibly important. And we have a article coming out very soon demonstrating the value of this. So in summary, hopefully I've shown you today how our liver MPS platform can produce very highly metabolically active and very stable, very consistent in vitro liver cultures. And these can then be used for a number of different applications, but particularly they can be used to understand human drug metabolism, looking at metabolic ID, or being used to study slow clearance compounds. We can use the system to look at drug-induced liver injury, and particularly we can really interrogate mechanisms of action and look at the details of how this injury works. And so as a company, we therefore want to make this technology available to really a wide variety of researchers. So hopefully, as you've seen, this technology is it's very simple to set up, and we make this technology broadly available to all different types of customers in academia and in pharma, and customers can either get this technology set up in their own lab, or they can also access it as a service through CM Bio. So just finally, I just want to mention there are 2 more webinars in this series in the coming weeks, which I encourage you to attend by colleagues at the University of Pittsburgh and the Massachusetts Institute of Technology, and I'm sure you'll hear more about these. Thank you.
Thank you very much, Thomas. I am sure you will all agree that there were some very excellent points raised during the presentation. We will now begin the Q&A session. To submit a question to our speakers, you should enter your question into the box on the right-hand side of your screen and click Send. We will answer as many questions as possible during the time available. We have had several questions submitted, so I'll move straight on to these. Firstly, how long does it take to induce DILI or any disease states, and for how long can you test thereafter using your system?
Thanks, Becky. Yeah, nice question. So I guess there's actually two questions in there. So the question is how long does it take to induce DILI and potentially induce a disease state? So we can use the system to study both acute and chronic DILI. So we can look at short-term acute responses over 24, 48 hours and mechanistically investigate that. We can also look at chronic dosing, for example, hepatocytes in our system for 2 or even 3 weeks, repeat low doses. So we can really ask a different set of questions. So it really depends on the experiment you want to set up. From a disease state perspective, generally setting up a disease state in the system takes a little bit of time, a minimum of somewhere around a week normally for example, to induce the fatty liver disease state that I showed there in one of those final slides. We've done other things with sort of viral infection, etc. These normally take a good few days up to a week to initiate, and then we can look to add therapeutics into the system. The system is very stable. We run our platforms generally for a couple of weeks up to a month, so we have a good window to do these kind of studies.
Brilliant. So second question, what is different about your solution that enables clinical measurements such as ALT, AST to be measured, as they are typically hard to detect in other systems?
Yeah, so we use a really wide range of endpoints to understand and detect DILI in the system. So we typically don't just use standard sort of ATP readouts that you might do in more simple systems. And one of the main reasons that we can detect sort of a really wide range of endpoints, particularly clinically relevant endpoints, is for 2 reasons. A, we have quite a large number of hepatocytes in our system. So the standard scaffold has up to 600,000 hepatocytes in it in a single culture. And we also have a relatively low cell to media ratio. And so that means the metabolites being produced by those hepatocytes in a relatively concentrated system, more so than what they would be in other standard sort of 2D in vitro setups. And so it allows us to have a more concentrated system that we can then go and detect the presence of these metabolites.
Brilliant. So another question, can this system use skin tissues and be used to test toxicity of topical transdermal drugs?
Yeah, absolutely. This is not an area we have placed particular focus to date. But yeah, the system is adaptable and can take a variety of different tissue structures and setups, particularly of barrier models. So barrier models typically being gut, lung, skin, even the kidney. So we can use the system to take a variety of different tissue types, and skin being one of those. We actually, as I showed you again, one of those latter slides, we have multi-organ setups where we could link something like a skin culture to a liver culture, and you could imagine how the local effects on the skin could then go on and affect other organs in the body. So we can set things up actually quite flexibly on the platform. But as I said at the beginning, sort of looking at skin toxicity is not a particular area of focus that we've looked at to date, and there are a number of other models out there that could do that.
Brilliant. And a question from one of our viewers is that they wanted to ask, how do you account for scaling of organs in multi-physiological systems, and do you have separate media for each cell type, or are they connected together with a common media, and how do you ensure that they all have their requirements met?
Okay, yeah, great question. Again, actually, there's 2 questions there. So one about scaling and one about common media. So to the scaling question first, is a really key aspect of any multi-organ MPS system, and something we actually think really very hard about. There are a number of ways to scale these platforms, and we don't have a sort of one-size-fits-all approach to this, where we standardize our scaling. We produce scaling metrics based on an application by application basis. Principally, our preferred scaling methodology is based around functional scaling. So how the function of organ A relates to organ B. Are they appropriate, and are the two cross-talking appropriately, rather than, let's say, more basic or more simplistic sort of allometric scaling approaches, which often come across significant challenges as you scale down the size of organs. So we think very hard about that, and we have a number of tools to do that. We have a team of in-house bioengineers with a lot of expertise in this space. And then the second half of the question was related to common media. So this is again a key thing that we've worked on a lot over a few years, and we think very hard about when we build methodologies and protocols for using multi-organ platforms. We generally can develop common media for multi-organ platforms. This often takes some optimization, and we think about how, for example, one organ might be phased out of its own media over a few days before it's brought onto the platform for the multi-organ study, rather than a sort of an aggressive switch between one media and the other. So yeah, there are different protocols and different setups for different multi-organ experiments.
Brilliant. And how does this model address immune-mediated IDILI, especially adaptive immune-mediated toxicology issues?
Yeah, so immune-related tox is a big challenge for the field broadly. So I guess we can do a couple of different things with our platform to try and address this. So one thing we can do, and I didn't show a lot of that in this presentation, but more than happy if the individual wants to reach out to us, I can share more information on this. Is we have a number of models where we incorporate immune cells into our individual tissue structures. These are often innate immune cells. So we have, for example, macrophages, etc., and we can use that to look at inflammatory-driven tox in particular organs. But the question actually asks about adaptive immune responses as well, and that's further challenging. But we have models whereby we can have, for example, mixed immune cells held within our platforms, for example, in different compartments separate from the liver tissue or the gut tissue. So those immune cells can crosstalk and interact with the different tissue structures, allowing you to potentially answer some of these questions about immune-related tox. But the adaptive immune tox space is fairly challenging.
Okay, brilliant. And how many endpoints readouts can you perform on each sample?
Yeah, so as I touched on earlier, we have quite a significant size to each individual well or tissue that we have in our system. So we have large numbers of cells in each tissue structure, but then we also have a significant amount of media. So we can typically repeat sample from a particular well multiple times to get longitudinal data about how a tissue responds, but we can also take multiple samples from the same well to get multiple endpoints. So in a liver DILI experiment, we can typically run 5 or 6 different endpoints fairly straightforwardly to understand both secreted markers, so for things like albumin, ALT, LDH, but we can also measure cellular-based markers such as caspase and ATP content, all from the same well, all at the same time. We think that's incredibly powerful. Get all of that data from a single microtissue that we generate.
That is brilliant. And unfortunately, that is all we've got time for today. So thank you very much, Thomas. Just to remind everybody, any questions that we didn't have time to get to will be answered offline as soon as possible. You can continue to ask questions even when watching this webinar on demand. A certificate of attendance is available to download from the handouts tab on the right hand side of the webinar platform. The webinar will now be made available on demand. If you have any friends or colleagues that you think may be interested in this webinar, please feel free to share the link. Thanks again for your time.